Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond. Welcome back to Neuroverse, and welcome to another Tools for Looking into the Brain episode, our mini-series sponsored by Scientifica, where we discuss neuroscience methods and deep dive into the world of academia. Today, we're joined by Matthew Harvey to talk about the cortex, calcium, and cognitive flexibility. Matt is a neuroscience researcher and currently a postdoctoral fellow at the Francis Crick Institute in London. Prior to this, he trained as a medical doctor, then made the jump to academia and carried out a PhD in neuroscience at King's College London in the lab of Adil Khan, where he investigated cortex-wide neural activity during learning and flexible decision-making in mouse models of neurodevelopmental disorders. He's currently working on impulsivity and cognitive control in Flor Iacaruso's lab at the Francis Crick Institute. Welcome to the podcast, Matt.
Oh, thanks for having me on.
So we normally start the episodes by just asking how you got into neuroscience in the first place. And of course, with your medical background, I'm quite curious as to what made you make that switch and choose this topic of research.
Yeah, absolutely. So I've sort of always been interested in psychiatric conditions. I think mental health conditions in particular can be kind of really devastating. And I think they can affect some of the core things that we think, make us ourselves and make us human. And so that was my initial motivation. So I wanted to go to medical school and be a psychiatrist. And I remember kind of learning about psychiatric conditions. And, when you learn about things like some cardiac conditions or infections, you can kind of get quite a nice sense for, okay, these are the mechanisms, this is the system. I can understand all the details and I can really imagine it. But I remember feeling pretty unsatisfied by all the textbook descriptions of psychiatric conditions. They always seemed a little bit hand wavy. Oh, there's maybe something to do with some neuromodulators like serotonin, but we don't really have any concrete kind of mechanisms or understanding of what causes them, because the brain is so complicated and we don't fully understand how the brain works. And I remember being, I think, in the third year of my medical degree and seeing like a news release of a paper from Misha Arons at Genelia, where they did sort of calcium imaging of the entire larval zebrafish brain. And I remember they had a video in the kind of press release and seeing all of the neurons firing in this brain. And I remember being amazed at the incredible tools and precision that was available in the kind of world of animal neuroscience. And then I think my experience, doing psychiatry in practice is that you kind of meet people who are really, really unwell. And you have, there's a small handful of different types of drugs. We don't have a really, really robust understanding of how any of them work. So if the patients ask me, oh, how does this medication work? I can't really tell them. If they ask me, okay, what is it that's going on in my brain that's causing this? It's difficult for me to give them a really concrete answer. And I can't really predict which medications are going to work. So we might have to try a few. And often they have really significant side effects that people can find quite difficult to deal with. So that really, I think, kind of inspired me, and I could see the incredible precision that we had in neuroscience and how useful something like that would be in the practice of clinical psychiatry. So that was always my dream, to kind of, can we use these incredible tools where we can see populations of neurons, individual neurons firing, we can manipulate their activity with light. Can we use, these incredible powers of mouse neuroscience to try and get a more mechanistic and a more robust understanding of how the brain works and psychiatric conditions, so that ultimately we can try and use that to get some treatments. Yeah, so then after did 2 years of medical training, I applied for the PhD position in Adil Khan's lab at King's. So when you're trying to find any insights into psychiatric conditions using the tools of mice, it can be quite challenging because mice don't necessarily have the same psychiatric conditions that people have. But over the past, I think, kind of 10 to 20 years, there's been a lot of progress in our understanding of psychiatric genetics. So there's a group of conditions called neurodevelopmental conditions that have a really high genetic component. So these are conditions like intellectual disability, autism, schizophrenia, bipolar disorder, and ADHD. And for things like autism, schizophrenia, and ADHD, it's kind of estimated between about 70 to 80% of the variance is explained by genetic causes. So in some sense, we kind of know what causes these conditions. It's genetics. When you actually look at the genetics, it is incredibly complicated. So I think there's over 900 genes that have been associated with autism, and they often interact and have many complex effects. So although you might know it's genetic, understanding how on earth does one person's genes lead to their brain working this way is a huge challenge. But the fact that it is genetic means that it's at least a way that we can access it and use the tools of mouse research. We can take some of these genetic variants that we find in people who have these conditions and put them into mice and see how does that change how the brain forms and how the brain works. And so that was what we did for the PhD. So there's a gene called neurexin 1 alpha, which is, we find that variations in this gene are present in people who have schizophrenia, autism, ADHD, as well as people who've committed suicide. And you can take these kind of mutations and we can put them into a mouse. And what we'd hoped to look at was how's the behavior of the mouse different and how's the neural activity change. And one of the things that Adil is really interested in and Adil's lab does is cognitive flexibility. So kind of how do I, flexibly change my plan and what I'm doing and my behavior as the environment changes. Humans and animals, the world is fast paced and changing all the time. You've got to be able to really adapt your behavior alongside with it. And we find this mutation in people who have autism. And one of the things that people who are autistic can really struggle with is change. So if something suddenly changes, that can be quite challenging, and they tend to have a preference for kind of predictability and reliable routines. And so we wondered if perhaps when we put this mutation into the mice, will they have difficulty with cognitive flexibility? And so Adil had developed this behavior where we switch between two rules. So I know you've had him on previously, but I'll give a brief recap of his behavior. But we get mice, we make them run on a wheel, and then we first show them one of two orientations of visual grating. And if they lick to one, they get a drop of delicious soy milk reward. And if they lick to the other, they get a timeout and they get no reward. And they'll do kind of like a block of, say, 40 trials of this. And if they do it well, then unbeknownst to the mouse, we kind of switch the rule. And so now when he runs, he'll still see one of the same gratings, but now licking has no effect. And then the grating goes away, and then we give them one of two odors. And one of the odors is rewarded. So if they lick, they get a drop of delicious soy milk. And the other odor is unrewarded. So if you lick, you get no soy milk. And so the mice have to kind of switch back and forth and remember what the rule is. Am I paying attention to what the visual stimuli are or what the odors are and what's important to me? And so we can see how mice flexibly change what they're paying attention to, what's important to them, how are they processing things throughout this behavior. And what we actually found is that the neurexin 1 alpha knockout mice were absolutely totally fine with cognitive flexibility. They were just as flexible, they could switch the rules, but they were really impulsive. So they found it really difficult to withhold their licks and not lick to the unrewarded stimuli. Yeah, so we had these neurexin 1 alpha mice, and we basically wanted to know how are they with cognitive flexibility, and are there any changes in the brain activity? So we kind of used this behavior that Adil's got in his lab to investigate how mice switch between different rules. And while they were doing that, we were measuring the brain activity with something called wide field calcium imaging.
Yeah, firstly, I always really enjoy hearing these origin stories, and I didn't realize that you had such a strong interest, like all the way from the beginning, all the way from medicine. And I think it's really amazing that you actually managed to like pursue that and answer the questions that you wanted to answer from the beginning. And as you mentioned, we have had Adil on. I also wanted to mention that to our listeners that if at the end of this episode you're interested in hearing more about the research in the Khan lab, and also we mainly talked about two-photon calcium imaging in the episode with Adil, which was episode number 75. So be sure to check that out after listening to this episode if you want to hear more. But Matt, I know that you focused more on wide field calcium imaging. So could you tell us a little bit more about what that is and what sort of information it gives us about the brain?
Yeah, so wide field calcium imaging in particular, it basically lets you zoom out. So when you're doing calcium imaging, neurons they communicate with these electrical impulses, but sort of alongside these electrical impulses, when they fire, they kind of fill with calcium, and so you can express these calcium-sensitive fluorescent dyes that basically will start to fluoresce more in the presence of calcium. And so you can use this to measure as a proxy for neural activity and you can kind of use whatever scale you like. And so, as you mentioned before, one thing that people do is they'll focus really tightly on one part of the brain, and then you can look at with really high resolution and see individual neurons firing. But behavior in the real world, it requires basically lots of different parts of the brain to work together in coordination at a really fast time scale. You need to coordinate activity across all your sensory regions to kind of perceive the world, across your motor regions to kind of output your motor decisions, and then across some of these prefrontal regions to modulate what kind of rules you're following and what's important to you. And then you've also got brain-wide changes in the state of the brain with things like neuromodulators, so things like serotonin and dopamine, as maybe the mouse is hungry or more aroused or withdrawn. So when you focus just on one particular brain region, you kind of miss the big picture. So what wide field calcium imaging is essentially it's putting a window over the entire dorsal surface of the skull. And one advantage the mouse has is that its brain is smooth, so there's no wrinkles, so you can kind of see the whole top part of the mouse cortex. And this means you get a zoomed out view, so you don't see individual neurons, but you get a kind of rough estimate of the average activity within a region. But it means you can kind of see across the cortex lots of these different regions being active. So we can see both the visual sensory regions as they're processing visual stimuli and the kind of motor planning regions as they're making their decisions and even like the primary motor cortex as they're executing their movements. And it also lets you see how these regions coordinate their activity. So I often think it's a really nice kind of first step when you're doing a behavior to zoom out and get the big picture and think, okay, these are the broad ways the brain is changing and the broad way these different brain regions are communicating to enact this behavior.
Yeah, I think that demonstrates really well like the different types of information you can get in neuroscience, and it can all be equally important, but in very different ways. Like sometimes you want to look at
Yeah.
the molecular mechanism, but sometimes looking at like crosstalk across whole regions is really important.
Yeah,
Yeah.
I also just wanted to mention that whilst you mentioned in the beginning, it's so important to look at the characteristics of all of these different developmental disorders and their genetics, the behavioral phenotype can be quite different. And of course, like across humans and animals, it's very different. But what I find interesting in animal behavioral research is that it can be quite stereotyped. And with that, I'm curious about a little bit more detail in terms of your behavioral paradigm and what you found and what brain regions were involved. So we now have an understanding of how calcium wide field works. Could you tell us a little bit about the brain regions involved at the important behavioral time points in your paradigm?
Yeah, absolutely. So one of the cool things you can do with this paradigm is you show mice these visual stimuli, and when they're following one rule, these visual stimuli are really important. They're kind of using this visual information to make that decision. But when they're in the other rule, you can show them exactly the same visual stimulus, but now they basically don't care about it, and they're not using this information at all. And so you can kind of see how does when the mouse switches its attention or whether something's important, how does that change how the information is processed across the brain? And so when you first kind of look, let's say you've got the rewarded grading pops up and the mouse reacts to it and licks, you see this kind of beautiful wave and propagation of activity across the cortex. So you start by getting these activation of these primary visual cortices, and then it spreads to these higher order visual areas, and then it kind of spreads medially into these motor planning regions, these kind of secondary motor cortex, and even along this kind of proximal to distal aspect of the secondary motor cortex. And then as it gets right to the kind of front of the brain, it kind of spreads more laterally to this region called the anterolateral motor cortex, where there's neurons there that kind of initiate a lick. And then as they start doing this lick, you get this activation of your primary motor cortex as they're kind of fine tuning these movements. And then when they're getting this kind of tactile feedback of the milk, the somatosensory and barrel cortex lights up. So it's really this delicate ballet of cortical activity that's active in response to when you're licking to something. And so one of the key things we found that's really cool is when you're paying attention to a visual stimulus and it's important, this activity propagates from these visual areas forward into these motor planning regions, into the secondary motor cortex. But when you're not paying attention, we still see activation of the visual stimuli, but it doesn't propagate further into these motor planning regions. So it's almost as if the brain is able to filter out what's important and decide where the sensory information is able to influence my current plan. And so when I'm paying attention, it's important. This information is able to influence other regions. And when I'm not paying attention, it's kind of filtered out.
But you mentioned that with
Yeah.
the neurexin 1 alpha mice that they would lick compulsively. So I don't know if you looked at wild type mice versus neurexin 1 alpha mice, and what was the difference in the brain activity? And did they still have that? Not only like it would light up with the visual cortex areas, but did it go on to the motor planning areas, or what was the difference?
Yeah, great question. So with the neurexin 1 alpha knockout mice, we found that when they're paying attention to visual stimuli and they're responding to them, we actually saw this really enhanced activity in the secondary motor cortex. So there's a ramp of activity that starts even before they see the visual stimulus, and then it kind of peaks much more rapidly and gets to a higher level. So they've got this what looks like kind of hyper excitable secondary motor cortex, which is maybe why they initiate movements kind of more rapidly and have a harder time stopping themselves initiating movements. But interestingly, that's only the case when the visual stimuli are relevant and when they're paying attention. When visual stimuli don't matter anymore, they're still able to filter out this activity. So it looks like maybe there's two different things going on there. They're able to switch their attention just fine and kind of filter the flow of activity depending on their attention just fine. But when something is important and it is tempting and there might be some soy milk reward involved, there's this just hyperactivity in the motor planning regions that I think makes it harder for them to inhibit their actions.
Really craving soy milk. Like every time I hear that in
Yeah.
these behavioral paradigms, I'm like, wow, soy milk has never sounded so tasty.
I know. Tell me about it. They'll run for 2 hours for little drops of soy milk. But I got curious and did taste it once, and it was pretty bland. I'm not sure. I'm not sure I'd be running for 2 hours for it.
Confessions of a Scientist. Um, I was curious on a more fundamental level because I think our listeners would have missed out on these hand gestures that were really adding to your
Oh, sorry.
description of the activity propagation. But I also remember seeing these videos of activity at the calcium levels in like one of your talks. But I was just thinking earlier, why does the activities propagate spatially? Because obviously activity in the brain is caused by a chain of connected neurons within neural circuits, which when you do the wide field calcium imaging, you're only looking on the cortical surface. So I guess you're looking at the more superficial cortical layers mainly. But there are also long range projections in the brain, and activity is obviously like entering from different neural circuits through different sensory systems. So I don't know if this is a bit of a strange question, but like, is the activity always like a wave of propagation across space, or can it also just be like different areas lighting up in the cortex?
Yeah, great question. So the answer, which is maybe a bit of a cop out, is it's kind of a bit of both. So you're right. So we see just the surface of the cortex, and underneath that, there's all these kind of subcortical loops going, to the striatum and to the thalamus. And so there's undoubtedly some of this propagation through these subcortical loops beneath the surface of the cortex that we can't see. And then also when you look at the cortex, so the majority of kind of cortical connections are local. So neurons tend to connect to neurons nearby, but there are some longer range connections as well. And so I think it's this mixture of the phenomena. I think you do have to some extent this literal spreading across local connections in the cortex like a wave, but then you also do have this long range routing through these subcortical structures. And yeah, I do wonder, one of my theories is you could use these routing through the subcortical structures to flexibly control how the cortical activity spreads. And I think that may be the process whereby you can kind of turn on and off how these regions talk to each other.
And did you test if preventing that additional secondary motor cortex activity in the mice that were more impulsive, in the neurexin 1 knockout mice, did you see if that could reduce the behavioral phenotype?
Yeah, so it's something we'd love to do. Unfortunately, we didn't have time to do that. And the other difficulty is we know from other research that if you inhibit that region, the secondary motor cortex, with something like optogenetics, then mice can't lick, right? So we did do that. That if you inhibit that piece of the cortex, they can't lick, but they basically can't lick at all because this is the region that initiates the lick. And so, you can look in neurexin mice, and I'm sure we'd find that if we inhibit this patch of the cortex, they can't lick. But in order to kind of tease apart, okay, what exactly is it that's going on? Is it this increase that's making them more likely to lick? I think just inhibiting that region totally is too much of a blunt tool. One of the things we did do is when we found this region that looked important, is we then did a little cranial window and did two photon imaging to see the individual populations of neurons to see if we can tease apart, oh, exactly, is it, are these pro-licking signals that are making them more likely to lick, or maybe it's some sort of compensation and there's some neurons here that are suppressing the lick. And that's something that we're still looking into. But yeah, unfortunately, we weren't able to cure the neurexins of their impulsivity. It's something I'd love to do in the future.
Well, I guess it also again shows that like the way that you've gone about it is actually a good way to kind of find something to then focus more on and try to understand maybe the deeper mechanism behind that.
So I can see that the visual cortex and the motor cortex are very involved in this behavior. But I was wondering if you could tell me a little bit more about the striatum's role, because that was also mentioned in the paper.
Yeah, so I think in the paper you're referencing, one of the people in the lab, Nick, he kind of put this microprism into the brain, which means you can kind of look directly at some of these prefrontal regions, which are normally folded away underneath the cortex that are really difficult to access. But he was able to image from these neurons within the anterior cingulate cortex, just a prefrontal part of the brain. And then he also deliberately labeled neurons that projected to the striatum, and then was able to kind of try and investigate to what extent do neurons that kind of project to the striatum versus neurons that don't project to the striatum contribute to this signal as mice switch between the two different rules. Um, and I think so. One thing that was really striking is that the current rule the mouse is in was really strongly represented in the activity within these prefrontal regions. You could almost just by eye look at the raster of neurons firing, and you could see what the blocks were. But there was also this kind of smaller signal as when the rule changes. Often mice are running, they're expecting an odor to come, and then this expected odor doesn't arrive, and there's a mismatch between their expectation and what happens. And we found there was a signal there in the anterior cingulate cortex that might be driving the mice to switch the rule.
That's very interesting. And so we've mentioned like these different brain regions. Did you look at any cellular specificity?
Yeah, so one thing we find is that with attention, the responsive neurons in the primary visual cortex change how they represent stimuli depending on whether it's something a mouse is paying attention to or not. And one mechanism we thought might be contributing to this is this class of neuron called VIP interneurons, and they're an interesting class of interneurons because they're thought to have this disinhibitory motif that they inhibit other inhibitory interneurons and therefore ultimately boost the activity of some of the excitatory neurons. And so it might be that if you're paying attention to something and you want to boost the activity of that representation in V1, you could do it via these VIP interneurons and maybe some top-down signals from the PFC, activate these VIP interneurons and disinhibit the neurons that would represent things you care about. So he did lots of experiments where he optogenetically activated and inactivated these VIP interneurons. But we found that the way that these neurons were modulating the network activity was actually totally different to the way that attention was modulating the network activity. So it looked like in this case, it probably wasn't VIP interneurons. There's a current postdoc in the lab, Philippe, who's looking, maybe it's PV interneurons. And so he's doing these very cool experiments where you can activate individual PV interneurons and see what's the effect on coding. And there's some very interesting results that he's getting that I'll let him talk about if he ever comes on. But yeah, so one of the cool things about kind of calcium imaging is that you can express these indicators in specific genetic cell classes. And so another part of the work in El's lab has been teasing apart these genetic cell classes. And yeah, perhaps PV interneurons might play a role in this.
When you do the wide field imaging, is the calcium indicator expressed throughout all neuron types? And could you do it where it's not expressed in all neuron types?
Yeah, good question. So when I did the series of experiments with the neurexin mice and the wild type controls, there it was just expressed in all neuron types, so just under a synapsin one promoter. But then I've done kind of other experiments where we were uh, doing the simultaneous imaging and optogenetic inhibition to kind of probe functional connectivity. And there we used this line that had the GCAP expression restricted only to CAMK2 expressing cells, which are predominantly excitatory cells. And that was good because that meant that we could turn on PV interneurons and we wouldn't get this interfering of turning on the PV interneurons and inhibiting the excitatory neurons and getting these signals blurred together. But you can absolutely do these experiments where you restrict it just to one cell type. So you could look at just the cortex-wide activity of PV interneurons or somatostatin interneurons or VIP interneurons. And when wide field calcium imaging was sort of first starting to be performed, there was a kind of series of papers of people doing this, just looking broadly at different interneuron classes. And there's an interesting paper from Simon Mussol from the lab of Anne Churchland, where he looks at different neuron types, but based on kind of either their layer within the cortex or their projection targets. So, looking at these kind of deeper neurons that project to subcortical structures versus these more cortical neurons. And he finds that there's these interesting subtle differences in the way that the activity propagates between these different projection-defined cell classes.
Very interesting. So I kind of wanted to bring it back to link what we've been talking about to the genetics that you mentioned in the beginning of neurodevelopmental disorders. So obviously this is a mouse model of one specific gene that's implicated, neurexin 1. And I was wondering, how do you think the mutation in this gene, which maybe you could also elaborate a bit more about what effect that has on the protein molecular level, but how could such a mutation lead to these changes in context-dependent brain activity? And kind of linking to that, the bigger picture question of just, do you believe that the many, many possible thousands of genetic mutations that contribute to neurodevelopmental disorders may converge on specific mechanisms, and at what scale are these at the like neural circuit level or whole brain or behavioral level or multiple?
Yeah, great question. So, yeah, focusing first on neurexin 1 alpha. So it's this synaptic adhesion basic molecule. And so most of the mutations that we find associated in people with neurodevelopmental conditions, it's a kind of loss of function of the gene. So you don't get as much or as effective synaptic adhesion proteins. And so what that tends to cause is you get kind of weakening or loss of some synapses. And it's expressed kind of throughout the brain. So you get these changes are really widespread. But one part in particular are some of these projections between the cortex and the striatum. And there's some interesting work from the lab of Mark Fusilio that's kind of implicates specifically some of these projections from these more kind of frontal cortical neurons to D2 expressing cells in the striatum. That you might have specifically weaker synapses here because some of the molecules that help anchor the synapse together aren't working as well. And these D2 cells of the striatum form what's called the indirect pathway. And basically, the overall effect of activating these cells is to inhibit the cortex and often to inhibit movement. And so these projections from the cortex to these D2 expressing cells might be used when you're trying deliberately to inhibit a movement. And so if this pathway is specifically weaker, your cortex might just not be able to inhibit some of these striatal neurons as effectively. And so you find it harder to stop yourself executing movements. This is one of the kind of theories and hypotheses. We haven't proven that yet, but this is one potential way that you might imagine this change in this gene leads to this effect in the molecule that then leads to this effect in the circuit that then ultimately has this behavioral phenotype. And then the extent to which genetic risk variants converge on a few pathways is really interesting. So when you look at neurodevelopmental conditions, they have a really strong genetic component. But interestingly, when you look at them, a lot of the genes are the same genes that increase your risk of autism, schizophrenia, or ADHD. It's not that there are specific autism genes and specific ADHD genes and specific schizophrenia genes. And also, many of these variants are found in the neurotypical population as well. So, the way in which you go from a certain collection of genetic variants to a certain neurotype is really complex. But it's also thought that there's probably some effect of the number and the kind of dose of mutations that you have. So maybe we all have, to some extent, a small number of genetic variants, and there's a kind of just a degree of genotype variation in the population. But as you accumulate more and more of these genetic variants, as you become more genodiverse, you probably become more neurodiverse. And it might be that if you have loads and loads, or some of these mutations with a really big effect size, you might end up with something like intellectual disability. But if you have a smaller number of mutations, you might end up with something like autism, and maybe even fewer less, something like bipolar. So when we look at these mutations, there's a few broad things they do. So many of them are involved in synapses. And others are involved in regulating the expression of other genes. So it's incredibly complicated. You have effects throughout the brain. But I can't help but wonder if what we really see is that as you accumulate these genetic changes, there are certain ways that the brain is just vulnerable, that it isn't really able to repair itself or rebound from. And as you accumulate these genetic variants, it tends to be in one of these ways that the brain will break or will start to cause problems. And so I sort of think that you'll have some convergence on your ultimate behavior because some of these systems might break more easily, but there's probably loads and loads of different molecular ways that you can get there. And I think when we look at things like intellectual disability or autism, when we look at things like social cognition, I wonder if they're affected so much just because these are some of the most complicated things that we do. And so in order to properly navigate the neurotypical social world is really challenging. And you need basically everything to go right. You need to not be overwhelmed by sensory symptoms. You need to be able to infer these very subtle social signals. And then you need to do this complex planning of how what I'm going to say is going to be interpreted and affected by someone else. And so just perhaps these social functions, yeah, they're the most complex thing we do, and therefore they're the most fragile. And they're the things that tend to break first. And so as you accumulate genetic variants, this is where you become different more rapidly.
Yeah, really interesting. And I think that really drives home the point of like the more that we've discovered about neurodevelopmental disorders, it's not like we've been able to solve them, so to speak, but rather that they have shaped our impression of how the brain works and how humans work and kind of like how we are cognitive beings. And
Yeah.
obviously, like this whole idea of neurodivergence and it being more of a spectrum, and it's not you have a disorder or you don't, but rather like the phenotypes can be so complicated and it can be such a large number of interactions. And yeah,
Yeah.
it's just very powerful to change our impression of understanding the brain through this way. Yeah.
Yeah, I think that's one of the things that I'm beginning to understand and take away more is that there's this rich variation in the way that neural circuits work, and I think it's more that there's just this rich variation in neurotypes and how we all are as people. And I think there's an extent to which in society we all tend to try and fit into a typical mold because then life is easier. But really, there probably is much more variation in the population in terms of neurodiversity than I think we expect. And I think when we see things like autism and ADHD, these represent just the extreme ends of spectrums upon which we all lie to some degree. And yeah, when you talk about solving them, I don't think that's a really useful paradigm to have. But I think we need to kind of think, well, there's certain things that if you have autism, there's certain things that are really hard and that you might really feel you'd benefit from if you could have something that might stop you getting sensory overload so much. That might be something that's valuable to you. So these are the things that we can try and tackle, the things that are really causing issues for the people who have these conditions. But there's some things that it's absolutely fine that you're divergent on. That if you have a few interests that you love really passionately, I think that's great. I don't think we need to be curing that or solving that at all.
Yeah, absolutely. I completely agree with that. But going back to one of your first goals into going into neuroscience, one of them being that you were receiving patients that you didn't know how to explain what certain drugs would do and what things were happening in their brain. I was wondering, now that you've seen a little bit of both worlds, the medical world and the academic world, what do you think we're missing in terms of research or perhaps a proposed experiment regarding attention or cognitive flexibility or impulsivity to be able to understand and start developing a therapeutic intervention when it is needed? Yeah. I know it's a very big
Yeah.
question. Feel free to, yeah, be as blue sky and abstract as you like about it, but I thought I would just pose that.
No, that's great. I love that. So definitely the thing that I've been kind of most surprised and taken away from the scientific work is some of this results from psychiatric genetics. And it makes me think that, when I was in the clinic, I perhaps naively tend to think, okay, well, there's autism and there's ADHD and there's schizophrenia, and these are three different things, and they probably have three totally different mechanisms. And everyone who has ADHD, probably it's the same thing going on in their brain. And I think that to some extent, that is the kind of underlying assumption that we make, right? If I'm doing a neuroimaging trial on ADHD, I get loads of people with ADHD and I scan their brain and then I look at the mean difference. And the underlying assumption is that therefore in this group of people, the underlying change in their brain is the same. But as we understand that more and more of these conditions are genetic and that the genetics are so overlapping and variable, it may well be that the genetic variants that have caused my autism might be entirely different from the genetic variants that have caused your autism. And so these things might not really be these distinct clinical entities as kind of unitary and simple as we thought. And it might be that there's much more of a kind of landscape of genetic variants and therefore a landscape of kind of neural circuit variants. And I think what I would really want to get at is to what is the meaningful kind of unit of change that people have. It might be that at the circuit level, there's a kind of a handful of these circuit changes which can be present to multiple extents across lots of different conditions. So, for example, I think it's about between 50 and 70% of people with autism also have ADHD. So many people have multiple conditions. And it might be that they've got these numerous circuit changes that when you have certain combinations of them can cause autism, other combinations can cause ADHD, and other combinations cause schizophrenia. So I would be really interested in trying to pin down specific circuit changes that might really be present across lots of different neurodevelopmental conditions and try and find what are these meaningful units of circuit change that people have.
And if we were to find those differences in the circuit changes, do you think that the interventions would be the gene therapy based? Do you think they'd be like electrical, like stimulations based? What would you predict?
Yeah, this is a really good and difficult question. So you're right. One thing you could think maybe perhaps naively is, oh well, these conditions are genetic, their genes are different. Why don't we just put the genes back? I don't know that that really is going to be the best way to do this because if anticipating the effect of changing one gene is really really hard, and to anticipate the effect of changing hundreds of interacting genes is going to be almost impossible. So I think that's probably not a very fruitful way, and you're going to have all sorts of consequences that aren't just ameliorating the symptom that you find distressing. But I think this is hopefully one of the nice ways that if you can really understand it as a system from the neuroscience point of view, you can get an idea of, okay, well, these are the ways that I can kind of modify that system. And so even if I don't change your underlying genes or your underlying circuit to be typical, if I give you this drug or if I give you this kind of transcranial magnetic stimulation or whatever perturbation, this will make the system work in a way that causes you less issue. So I think going forward, I think it's more about trying to characterize the system and understand exactly the ways that drugs or any of our current interventions impact this system, and then trying to predict, therefore, that will be useful for this subgroup of patients rather than any development of something like a gene therapy. I mean, there are, for things like epilepsy, I think there are gene therapy trials underway. There's maybe some conditions like epilepsy where that is the way forward, but I think we're a very long way for anything like a neurodevelopmental condition.
Yeah, thanks. Really interesting insight.
Yeah, thank you for attempting to answer those really difficult questions from apparently.
No,
Um,
no worries.
well, I think that's a great place to wrap up today's episode then. Thank you so much for joining us today, Matt.
Yeah.
No, thanks for having me.
Yeah, thanks for letting me pick your brain.
And thank you for listening to today's episode. And thank you once again to Scientifica for sponsoring the Tools for Looking into the Brain series. Do check out their website, scientifica.uk.com, for more information on electrophysiological systems and tips and tricks. And for more information about Neuroverse, check out our website, neuroversepod.com, and follow us on social media at neuroverse_pod.